Friends, I am Dr. K.D. Mishra and today we will discuss immunity. Immunity is the capacity of multicellular organisms to resist harmful microorganisms, that is pathological organisms, which creates some kind of disturbance in our normal physiology, and they are disease causing organisms. So the ability of body to defend against invading agents is called immunity. So either a pathogen directly enters into the body or some kind of other proteinous substance called as antigen enters in our body, which is harmful to us. It is dealt with a defense system that is called as immunity. Now, by developing and maintaining immunity, we protect ourselves from various diseases. The immune response of our body involves two important steps. The first one is, and the most important, is the recognition of pathogen or antigen, any foreign material that enters into the body. So first is the recognition of the foreign antigen or pathogen, and second one is to destroy it, the reaction to eliminate it. Now the classification of immunity. Now broadly, immunity can be categorized into two categories. One is innate or non-adaptive immunity. Non-adaptive immunity means the immunity which we get from the parents through genes. So it is a kind of genetic immunity that is provided with the genetic constitution of an individual. It is since birth, it is by birth, and it remains throughout the life. The second one is acquired immunity or adaptive immunity, the immunity which we acquire after birth as we are exposed to different kind of diseases and pathogens. We acquire immunity against them. So it's an adaptation to survive better against the invading pathogens. Therefore, it is called as adaptive immunity, and since it is acquired after birth, it is called as acquired immunity. Now, this is further classified into naturally acquired immunity. When we say naturally acquired immunity, that means at some or other time of our life, we are exposed to some disease causing organisms or disease causing materials. So when we are exposed to that, we develop an immunity naturally. This is called acquired immunity. Now, the other one is artificially acquired immunity. If we acquire the immunity after artificial entry of pathogen or antigen into the body, that is called as artificial acquired immunity. Now, this can further be classified into two categories. One is active artificially acquired immunity and another is passive artificially acquired immunity. We will discuss this later, but for the time being, the active immunity is that which is provided by some kind of vaccine which is given. Then the person or a child develops acquired immunity against it, and it works for almost throughout the life, because for that our system generates a memory, and that memory is there, and whenever there is a infection by the same pathogen next time, then the immune action is immediately evoked and it works. On the other hand, the passive artificially acquired immunity is that when the antibodies or anti-sera is given to fight with that particular pathogen. This is, although it is fast, but it does not work for a longer period, because the immunity is developed outside and the antibodies or anti-sera is injected so as to provide the immunity. Now, here again a classification is given in more clear way. Now, the acquired or adaptive immunity we divide into two: active immunity through antigen and passive immunity through antibodies. When the immunization is done, then it is called active immunity. Either a person is directly infected by the pathogen and then it develops antibody against it, then it is called as natural immunity, natural active immunity. And if the pathogen is given in the form of vaccine, you know, in vaccine what we do is that we take the pathogen, either virus or any other, we make it ineffective, it is attenuated, its disease causing capacity is destroyed, and a vaccine is formed that contains the protein of the same disease causing organism. So, that forms the vaccine. When this vaccine is injected into a person, he learns how to develop antibody against it. So, this is gained by artificially by vaccination. Now, in case of passive immunity, you see, one is the maternal immunity, that is the immunity provided by the mother to the child. It can be provided through placenta during the intrauterine life. The antibodies are able to cross the placental barrier. You know, when there is some infective agent, the mother is first of all affected by that. The mother develops antibody against it, and those antibodies crosses the placental barrier and reaches into the fetal circulation, thereby protecting the embryo developing inside. Now, then after birth, in the postnatal life, you know, it has been advised always that the baby should be fed with the mother's milk for first few months, maybe about 6 months, because mother's milk contains antibodies, and those antibodies prevent infection in the neonates in the postnatal life. Then passive kind of immunity is provided by using some kind of anti-sera that contains antibodies in the serum. We have already explained serum is a part of blood from which the formed elements are removed, the fibrogen is removed, The remaining part of the blood which is transparent is called serum, and when this serum contains antibody, it is called as antiserum. So, if a disease is there and it has to be dealt with immediately, like in case of tetanus, if there is an infection, there is a possibility of an infection, and some time is spent before taking this vaccine, then antiserum is given. This is called as anti-tetanus serum. So, it works immediately, but such type of passive immunity is retained only for a limited period. Now, coming on to the innate immunity, that is the immunity which we have since birth. The first part of it is non-specific defense system. That means the defense of our body against any organism that enters into our body. It is irrespective of the type of pathogen. That means it is not specific for a particular type of pathogen. Whatever organism enters into the body, it is dealt with. So, non-specific defenses, it includes skin. It is the first barrier between disease causing organism and our body. Then mucous membrane. Mucous membrane is provided on all the internal surfaces of our body. Now, these two provides mechanical and chemical barriers against the invading organisms. Then there are certain antimicrobial mechanisms like interferons which acts against viruses. Then there are certain complements. Then there are natural killer cells which kills the pathogens, whatever kind they are. Then an important phenomena in our defense system is the phagocytosis. Whatever pathogens enter into our body, they are engulfed by phagocytes, they are digested by them and destroyed by them. Then there is another important phenomena involved in our non-specific defenses. It's the inflammation and fever. You know, whenever there is an infection in a part of our body, the part of our body gets swollen, it becomes reddish because of dilation of blood vessels. Lot of our WBCs are attracted there. So, this phenomena is called inflammation, and ultimately the phagocytes kills the pathogenic organisms which are accumulated there. Such non-specific defenses can be further categorized into first line and second line defenses. First line defenses are those which deals with the disease causing organism at first instance, and second line defense are those which deals with the pathogens in the next stage. Now, here in this picture you can see the non-specific defenses of our body. See, we starting from here. In the eyes, there are lacrimal glands which secretes tears. Tears contain lysozymes. So, whatever pathogenic organisms enters through our eye, they are destroyed by the lysozymes present in the tears. Then there are lysozymes in our saliva. Another important entry point is our mouth. So, whatever pathogen enter from the mouth, they are killed by the lysozymes in saliva. Then in bronchial passage, there is a mucus membrane. It is a pseudo-stratified, ciliated mucous epithelium which traps all the microorganisms that enters through the inspired air and then kills them and then throws them out through the nasal passage. Then, in the stomach, the parietal cells in the gastric glands of stomach, they secrete high concentration of HCl that kills the pathogens. It is assisted by the enzyme pepsin. It is found in the stomach in the form of pepsinogen, which is converted into pepsin by HCl. These two combinedly kills the pathogens entering into the alimentary canal. Then when the chyme from the stomach enters into the duodenum, there is a sudden change of pH from acidic pH to alkaline. So by that sudden change, the pathogens are destroyed. Then there are, if some microorganisms develops in the urinary passage, they are flushed off from the outflow of urine. In case of females, the pH of vagina is acidic and that inhibits the growth of microorganisms. So that is how the first line defense system in our body works. Here is the skin, which provides a physical barrier. There are sweat glands with lysozymes. There are sebaceous glands which contain fatty acids. They are microbial in nature. Now here again the first line defenses, as I described just now. The skin, the superficial layer of skin, that is the epidermis of skin is keratinized and it resists the entry of bacteria. The acidity of skin is about 3 to 5, which inhibits the growth of bacteria. Then sebum contains fatty acids, which is bactericidal in nature. Then mucous membrane of stomach provides a mechanical barrier against the entry of pathogens. Then just now I explained that HCl and pepsin kills the bacteria which enters with the food. Then pseudo-stratified ciliated epithelium of the respiratory tract. It deals with the foreign elements, pathogens and dust particles and others. They are trapped in this mucous epithelium and with the help of ciliary movement, these are thrown out of the body. So that's how these first line defenses work. Now coming on to the second line defenses. When the pathogen gets an entry into the body crossing the first line defense, then they are dealt with the second line defense system. It is non-specific cellular and chemical defense system. The one is phagocytosis. It is the most important defense mechanism of our body. The microorganisms are phagocytized by phagocytes present in various organs. They are digested by proteolytic enzymes. This is the most common second line defense system. In almost each and every organ of our body, there are cells which have the capacity of phagocytizing the foreign organisms. So irrespective of what kind of they are, what kind of antigen they have, if they are foreign to our body, they will be engulfed by the phagocytes. So phagocytic system is there which takes care of foreign elements entering into our body. These phagocytes include Kupffer cells in the liver, alveolar cells in the lung, histiocytes present in the lymph nodes, osteoclasts present in the bone, similarly chondrioclasts present in the cartilage, the microglia which are present in brain, mesangial cells present in kidney, macrophages of spleen and lymph nodes. So, all of these cells, they are phagocytic in nature and plays a major role to deal with the disease causing microorganism that enters through the first line defense into our different organs. Now, here in this picture, you can see the mononuclear phagocytic system. As we have just seen, there are microglial cells in the brain, there are alveolar macrophages in the lungs, skin has Langerhans cells, then spleen has macrophages, the kidney has mesangial cells, in the liver Kupffer cells are there, in the blood monocytes are there, we have already studied, they are phagocytic in nature. Then lymph nodes are there throughout the body, they have macrophages which filters whatever pathogens enter into the lymphatic system. So, there are lymph glands at the strategic points of our body. There is a chain of lymph nodes here called inguinal lymph nodes. There is a chain here, 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 cervical lymph nodes, auxiliary lymph nodes, then inguinal lymph nodes. So, lymph when returns into the blood, it passes through a chain of lymph nodes where macrophages are there, which filters and destroys the pathogens, thereby saving us. Now, apart from macrophages, there are natural killer cells called as NK cells. These cells are large granular lymphocytes, they are modified lymphocytes, they react non-specifically and kill virus infected and cancerous cells by releasing perforins and cytolytic chemicals. You know, when a virus enters into our body, it is very difficult to kill that. Therefore, the natural killer cells, they kill or they destroy the cells which are infected with virus so as to check the reproduction of virus. Similarly, the cancerous cells, they have the tendency of multiplying very rapidly in an uncontrolled manner. So, those cells are to be identified and they have to be destroyed to check the cancer. This is done in two ways by NK cells. One is by secreting perforins. Perforins are the chemicals which forms pores in the cell wall of pathogen or a cell affected by virus or cancerous cell, by which, you know, when there are perforations, water enters into the cell, thereby causing destruction of the cells. So, this is one way. Another way is by releasing cytotoxic chemicals, the chemicals which are harmful to the cell. So, by these two methods, the natural killer cells kills the virus infected and cancerous cells. Third one, and the most common defense mechanism observed commonly by a common person also is the inflammatory response. In this, there is a role of macrophages. You know, as soon as there is an infection at certain place of our body, then what happens? The microorganism multiplies very quickly locally there, and a lot of fluid accumulates there. The blood vessels are dilated, a lot of macrophages from the blood are released there. The mast cell releases the vasodilators, then WBC and chemicals are released. So, all these accumulate in one place, that is called as inflammatory response, along with increase in body temperature. So, this saves us. This is the primary method of saving us from the infection. It occurs in 4 main steps. Now, steps in the inflammatory response involves, first one is leukocytosis. As soon as there is an infection, neutrophils are produced in the bone marrow and they are moved toward the site of infection. Then margination, that is neutrophil adhere to the wall of capillaries of injured tissue. So, wherever there is an injury, the neutrophils are migrated there and they becomes attached to the wall of capillary there. Then there is a phenomena called diapediasis. You know, diapediasis is a phenomena. Suppose this is the wall of capillary, then and this is the neutrophil. Now, what happens? This neutrophil forms a pseudopodia here, and then through this pseudopodia, the neutrophil comes out to the site of infection like this. So, this phenomena, by forming pseudopodia, when the neutrophils come out of the capillary wall, it is called as diapediasis, and there then the neutrophil phagocytizes the pathogens and destroy them. And the fourth one is chemotaxis. In this process of inflammation, certain chemicals are released which attract the neutrophils to the site of infection. So, this is called as chemotaxis. Chemo means chemical, taxis means movement, a movement of a cell towards the some kind of chemical is called as chemotaxis. So, by chemotaxis, neutrophils are attracted. All these works together. You know, as soon as infection is there, chemotaxis is there, diapediasis occurs, lot of leukocytes are produced, and then there is a margination, they are attached to the capillary wall, and then they are excluded from the capillary wall through diapediasis, and this goes on. This is how the inflammatory response occurs. Now, here in this picture, you can see steps in inflammation. There is vasodilation, then capillary permeability increases, then there is phagocytosis by neutrophils. Then in the final stage, that damage to the tissue has to be repaired, and for that there are fibroblast cells in the tissue which secretes growth factors, which helps in repair of the tissue. Here in this picture you can see, as soon as there is an inflammation, a vasodilation is produced in the endothelium of capillary wall. the neutrophils, they extrudes through the forming pseudopodia. They comes out at the point where infection is there. At the same time, the macrophages and fibroblast cells releases certain growth factors that helps in repair of the damaged tissue at the site of inflammation. Now, anti-microbial proteins. These have a specific role in controlling the reproduction of microorganisms. You know, when the microorganisms enter into the body, their special characteristic is that they multiply very fast. So, if we can check the multiplication or reproduction of microorganism, then disease can be prevented. So, the one type of protein involved is called interferon. It is very important. It provides primary defense mechanism against viruses. When one being infected by the virus, the body cell produces interferons, which stimulates other cells to produce proteins that interfere with the viral replication, that is the multiplication of virus. Now, what are these? The lymphocytes produces gamma interferon. The leukocytes produce alpha interferon, and fibroblast cells present in the tissue produces beta interferons. Now, the second is the complement. These are a set of about 20 proteins which are activated by foreign antigens. These activate inflammatory response and causes lysis of pathogens. Now, coming on to the adaptive or acquired immunity. Adaptive or acquired immune system is a functional system which recognizes the foreign pathogen or antigen and acts to immobilize it, then neutralize it, or completely destroys it. So, that is how the adaptive acquired immunity works. That is, first one is the recognition of pathogen or antigen, and then second one is immobilization or destruction of it. It is antigen specific. That means a specific kind of immune reaction is generated for a specific kind of pathogen or antigen, and it also stores memory to generate immune response whenever the same pathogen again infects that individual. It is of two types. Here you can see the acquired immunity is broadly classified into two types. One is humoral acquired immunity, which is mediated by production of antibodies. So, an immunity which is dealt with production of antibodies is called as antibody mediated immunity or humoral acquired immunity. The second one is when cells are involved in the providing immunity, then it is called as cellular immunity. Mostly T cells are involved in it, therefore it is called as T cell mediated immunity. So, broadly acquired immunity is divided into two. in one which is provided by antibody called humoral antibody or antibody mediated acquired immunity. The second one which is provided by a specific kind of cells called T cells, therefore it is called a T cell mediated acquired immunity or cellular immunity. Now coming on to cellular immunity. In cellular immunity, the cells are responsible for killing of pathogens or foreign antigens. The cytotoxic cells are involved, which are called as killer T cells or simply killer cells. Previously, we have studied natural killer cells, which are non-specific. But these cytotoxic killer cells, they are T cells, they are formed for a specific antigen. So, it is a specific immunity against a pathogen. These cells kills the pathogen. Now, the cellular immunity provided by these killer T cells is effective against intracellular organisms. This is important. The cellular immunity is affected against intracellular organisms, means those pathogens which enter into our body cells. So, it is effective against them, whereas humoral antibody is mostly effective against the extracellular organisms. We will see it later. So, it is effective in cases of viruses, parasites, intracellular parasites, fungi, tumor cells, and against transplanted tissue. If some tissue is transplanted that doesn't suit our body, then it has to be destroyed because it contains foreign antigens. So, cytotoxic T cells or killer T cells, they provide cellular immunity against various infections. Now, the humoral immunity, as I said just now, it is an immunity provided by antibodies, which is also antigen specific immunity for a particular pathogen or a particular antigen. A particular antibody is formed that provides the immunity. Those antibodies attach with the antigen and kills it. The antibodies are produced by plasma cells. That's an important thing. You know, the B cells, that is Bursa equivalent cells produced in the bone marrow, they mature and transform into plasma cells. The plasma cell produces antibody for a specific antigen. Then humoral immunity is effective against, this is important, extracellular organisms. The cellular immunity was effective against intracellular organisms. This is effective against extracellular organisms, especially in cases of bacterial diseases. And if some antigen enters into the blood or body fluid, then it is dealt with humoral immunity. Now, development of immunity. You know, when a specific antigen enters in our body, it stimulates development of a specific set of lymphocytes. You know, lymphocytes are produced primarily in the bone marrow, then they are transferred into different lymphoid organs. So, primarily it is produced by the lymphoblast cells, in bone marrow, then they migrate into primary lymphoid organs. There are so many lymphoid organs in our body, and there they multiply and mature. So, lymphoid organs always possess specific type of lymphocytes. Now, some of these lymphocytes which reaches into thymus, they mature there and called T lymphocytes. And those which mature in bone marrow, they are called as B lymphocytes. So, B lymphocyte mature in bone marrow and fetal liver, whereas T lymphocytes matures in the thymus. Here, the T word is suggesting thymus. Mature lymphocytes are released in the blood and they migrate into secondary lymphoid organs. Like lymph nodes, as we have just seen in the figure, there are lymph nodes at various strategic positions of our body. In the neck region, the axillary region, in the inguinal region, there are lymph nodes. So, these lymphocytes settle there in the spleen, in the Peyer's patches. As you know, the large intestine contains lot amount of lymphoid tissue called Peyer's patches. So, all these settles there for future action. Now, here in this picture, you can see how the immunity develops. Now, in bone marrow, there are lymphocytoblast cells which forms lymphocytes. These lymphocytes form pre-T cells and pre-B cells. Pre-T cells reaches into thymus and matures into T4 cells and T8 cells. We will discuss them later. And B lymphocyte, they mature in bone marrow. These are bursa equivalent lymphocytes. These B lymphocytes are converted into plasma cells, and plasma cells produces antibodies, and these antibodies provide humoral and immunity. Here in this case, cellular immunity, the lymphocyte will mature in the thymus, the T4 cells and T8 cells. They are cytotoxic to antigens. They kill antigens and provide cellular immunity to us. That is how the whole immune system works. Now, regarding the development of T cell, as I just mentioned that the T cell mature and develops in thymus, that is in the medulla part of thymus. Thymus is larger in the children and infants. As the advancing age, with the advancing age, the thymus becomes atrophied and it becomes fibrous in the adult stage. So, whatever T lymphocytes are formed, that are formed in the infants and children's. Lymphoblasts differentiate and proliferate into pre-T cell and finally into T cells. Now, during the development of lymphocytes, especially T cells, T lymphocytes, there are two changes, morphological changes and chemical changes. Among the morphological changes is that they develop specific peptide receptors for reception of the particular antigen. So, they are called T cell receptors. So, first of all, these receptors are formed for a particular antigen. Then, synthesis of chemokines. The T cells synthesize chemokines which are capable of killing invading organisms. Important chemokines are lymphotoxin and interferons. So, in this way, the T cell works. First of all, they form the receptor sites for receiving a particular pathogen or antigen. This is antigen specific. That means a particular receptor protein is formed for a particular antigen. And second is the release of chemicals which is helpful in killing the pathogen, which includes lymphotoxins and interferons. Now, types of T cells. There are three types of T cells. Number one is helper T cell or T4 cells. They are called helper or inducer as they help in induction of cellular and humoral immunity both. The T4 cells, they induce both type of immunity. They also induce the production of antibodies as well as they also induce the cellular immunity. So, therefore, they are called as helper cells. They help to develop immunity in both the mechanisms. They contain CD4 protein on their surface. Therefore, they are called as T4 cells. The T4 terminology comes from this CD4 protein. The second is cytotoxic T cells. Those T cells which kill the foreign antigen or foreign pathogen, they are called as cytotoxic T cells. They are called as T8 cells because they contain CD8 protein. Therefore, they are called as T8 cells. They are also called as killer cells because they kill the invading organisms by cytotoxicity, means having toxic substances that kill the pathogen. And third one, and most important in the process of immunity, is the memory T cells. You know, at some point of time, a person is infected by a disease or vaccination is given to him as a prevention of that disease. So, when first time it is given, specific cells are produced. Those cells retains the memory how to form antibodies in future when second time the same infective agent enters into the body. Now, these memory cells retains the memory almost throughout the life. They are long-lived cells. A small fraction of T cells remains in tissue as memory T cells. They have a very long lifespan, maybe entire life. These are capable of producing millions of T cells instantly. What happens? When first time infection is there, development of immunity takes about 10 days. But when he is infected second time, the immunity develops instantaneously because of these memory T cells, and it develops within a small period of 2-3 days. Now, B lymphocytes. B lymphocytes are called bursa equivalent lymphocytes. You know, in case of birds, near their cloaca, that is a sac which opens in the cloacal opening like anus. So, these cells develop there in the birds. So, the name comes from there. And they develop in the bone marrow. Then they are shifted into the lymphoid tissue. During the development of B cell acquire receptors. They also acquire receptors to recognize antigens and genes for immunoglobulin synthesis. That is the important part of B lymphocytes, that they develop receptors for a particular type of pathogen or antigen. At the same time, they develop genes for producing immunoglobulins, that is antibodies for that particular pathogen. Each B cell lineage is committed for making antibody against a specific antigen. That is, for each disease causing organism, a specific antibody is formed. Now these mature B lymphocytes, they migrate into the lymphoid tissue and stored there. They are stored in lymph nodes, in bone marrow, in blood itself, in other tissues. These transforms into plasma cells. And plasma cells are responsible for production of antibodies. So B cells are responsible for production of antibody through development as plasma cells. Now these antibodies either kills or neutralizes the antigens or pathogen. Here again, a small number of B lymphocytes, they forms memory B cells. These cells retain the memory how to form antibodies for that particular antigen or pathogen, which persist in the body for many years, and whenever there is a secondary infection by the same pathogen, these memory cells instantaneously produces large number of antibodies. Now the antigens have two important properties. One is the immunogenicity, that is ability to provoke immune response by production of antibody and proliferation of T cells or both. So, an antigen is that which stimulates or provokes the immune action in the body of a person. Then second characteristic of the antigen is the reactivity. Its ability to react with antibody or a cell. In case of humoral immunity to the antibody, in case of cellular immunity to a cell, or both. So, a antigen is that provokes the immune action and then chemically it is reactive against a particular type of cell or an antibody which destroys it. Now here is a specific term called MHC antigens, that is major histocompatibility complex, MHC antigens, or they are called as HLA antigens, that is human leukocyte associated antigens. These are self antigens which help in identifying and rejecting the foreign antigens. So, formation of MHC is very important in the process of immunity. They are divided as MHC1 antigen. They are found on the cell membrane of all body cells except RBCs. So, MHC1 type of antigens, they found on the membranes of all body cells except RBCs, whereas MHC2 antigens are found on the antigen presenting cells called as APCs, thymus cells and activated T cells. So, these are two types of MHC antigens, MHC1 and MHC2. Now, mechanism of cellular immunity can be summarized into 3 steps. Number 1 is antigen recognition, processing and presentation. Now, first of all, the antigens are recognized, then they are processed and they are presented to the cells, immune cells. On entry in the body, the antigen activates B cells by binding with the receptors on its surface. So, this is the first step. Now, the APCs, that is antigen presenting cells, the cells that present and process antigens to T cells, are called antigen presenting cells. These are B cells, macrophages and dendritic cells. Now, there are 5 steps in the process of antigen presentation. Number 1 is ingestion of antigen by APCs. The antigen presenting cell ingests the antigen by endocytosis, by just engulfing the antigen or pathogen. Then it is being digested, then formation of vesicles after digestion of antigen. Due to this, the peptides of antigen are broken, the fragments are formed. Simultaneously, MHC, that is major histocompatibility complex, MHC2 molecule are formed. Then the fusion of vesicles with MHC2 molecule. Those vesicles which are containing peptides, they are fused with MHC2. Then MHC-Ag complex is formed by the fusion of vesicles with MHC1. That is, the antigen plus MHC, they form MHC-Ag complex. Then incorporation of MHC-Ag complex into the cell membrane of APC. Now after this digestion and formation of this complex, this MHC-Ag complex is attached to the cell membrane of APC. Such APC attaches with T cell receptor and then activates the T cell. That is how the antigen presentation occurs. Now, the second process is when the TCRs, that is T cell receptors, come in contact with MHC-Ag complex, which is present on the APCs. So, APC attach to the T cell at the TCRs. These T cells are now activated. The activated T cells are stimulated by helper T cells, that is T4 cells, and then they differentiate into cytotoxic T cells, which kills the pathogen or antigen, and some of them are converted into the memory T cells which retains the memory for future. Then cytotoxic T cells or killer T cells which contain CD8 protein are called as T8 cells. They contain the proteins which kills the microorganisms. So, therefore, they are called as cytotoxic T cells. That is how the antigen is presented by the APCs to T cells, and T cells starts immune action. Now, finally, the elimination of invader, that is elimination of pathogen or antigen which is harmful to the human body. Now, this includes three processes. One is cytotoxic. The killer cells, they secrete perforins, as I explained earlier, due to their formation of pores, the certain chemicals which dissolve the cell membrane of pathogens, and from there water enters into the cells. So, water enters into microorganism and finally causing osmotic lysis. That is lysis or destruction of cell due to entry of water. Then secondly, they releases lymphotoxins. Lymphotoxins secreted by T cells kills microorganisms. Then interferons, as you know, in case of viral infections, interferons are produced. T cell secretes gamma interferons which are antiviral in nature. So, they prevent from the viral infection. Now, where a specific type of immune action is effective. Now, first of all, we take on cellular immunity. It is effective against intracellular pathogens. In case of viral infection, when virus enters into the body cells, those body cells are destroyed. In case of fungal infections, in case of chronic bacterial infections like tuberculosis, brucellosis, in case of certain parasitic infections, for destroying tumor cells, especially cancerous cells, and for destroying the transplanted cells if they are not suitable for the body. So, therefore, cellular immunity is effective against intracellular pathogens specifically. Now, mechanism of humoral immune system. It involves 5 basic steps. Number 1 is, as we just discussed, presentation of antigen to the particular immune cell. This is B cells serves as APS, that is antigen presenting cells, for developing humoral immunity. Then activation of B cells. Antigen binds to the receptor of B cell. On this surface, this process is accelerated by type 2 helper cells. As we have already mentioned that helper cell helps in accelerating this process. Then those activated B cells finally produce plasma cells and some memory cells, and those plasma cells forms antibodies to provide humoral immunity. Here again the same thing is mentioned, differentiation of B cells into plasma cells. They are transformed into plasma cells containing large amount of cytoplasm with more amount of rough endoplasmic reticulum. T helper cell stimulates this transformation process. Then proliferation of plasma cells and production of antibody. Plasma cell proliferates into millions and synthesized large quantity of antibodies. Here in this flow chart, you can see the same chain of reaction when there is a specific antigen infecting the person, then specific B cells are produced. Those B cell produces plasma cells, which are clonal because a single type of B cell is produced for a specific antigen. So, all those cells produced from that B lymphocyte are similar genetically, so called as cloned plasma cells, and cloned plasma cell produces antibody which are specific to that particular pathogen. Now, finally, killing of invaders or pathogens. The processes involved in this destruction of pathogens is number one, neutralizing the antigen by neutralizing bacterial toxins to prevent immune reaction. So, first of all, the antigens are being neutralized. Then, if they are not neutralized, then immobilization of microbes. They restrict the spread of microscope to other tissues, as when there is an infection of bacteria at one part of the body, by immobilization they are restricted to that part and they are prevented from being spreaded to other part of the body. Then activation of complement system. Now, this is a system of toxic proteins which helps in protecting from the invading organisms. The antibodies activates classical pathway of complement proteins. These are plasma enzymes which kills the microorganisms. Next one is the precipitation of antigens. The antibodies attaches to the antigen causing agglutination and precipitation of antigens. So, they are being clumped with the antibodies and they are killed. Then felicitation of phagocytosis. In this process, the phagocytes are stimulated, they are attracted towards the side and they kill the pathogens. The antibodies makes the antigens tasty. By the action of antibodies, the taste of the antigen is improved, and by that the phagocytes are attracted towards the antigen. This phenomena is called as opsonization. Now, coming on to the immunity to newborns, IgG antibodies can easily pass from the maternal blood to fetal blood through placental barrier. This is important. IgG type of antibody is capable of crossing the placental barrier from the mother to the fetus, and it provides passive immunity to the fetus. Then IgA antibody, another antibody which passes from maternal milk to the newborn, that also provides a passive immunity to the newborn against various kind of diseases. Now, as we discuss memory T cells, certain B cells also retain the memory for production of plasma cells and in turn production of antibodies. A small number of activated B cell becomes memory cells. These are long-lived cells. Sometimes they are retained throughout the life. These respond quickly and vigorously in large numbers to same antigen on subsequent exposure, that is called secondary immune response. Because when the same pathogen causes infection, next time these memory T cells generates the immune action very quickly. So this is called secondary immune response. Now we can classify the immune response into two basic categories. One is the primary immune response, that is when first time the pathogen infects a person. So a response is induced on first exposure. Antibodies are formed, but they are formed slowly. It starts formation within 3 to 6 days because first of all the cell learns how to produce antibody. There is a programming for producing a specific kind of protein which functions as antibody. Then when the same pathogen again infects a person, then the antibody are generated very quickly, but first time the peak level of antibody formation is achieved only after a long time, that is about 10 days. Then the number of antibodies in the blood declines gradually, but memory cells are there which retains the memory. IgM and IgG antibodies are formed in primary response, mainly IgM type of antibodies. Now when the same pathogen, same antigen infects second time, the secondary immune response is generated. It occurs especially and intensely, speedily and intensely. It is very quick due to memory B cells and memory T cells. Antibody level reaches at the peak within 2 to 3 days. There we have seen the peak level of antibody reaches at about 10 days in case of primary infection, primary immune reaction. But when it happens second time, it generates very speedily and quickly within 2 to 3 days, and they remain for longer duration. The secondary response main antibody type is of IgG type. Now antibodies, or they are called as immunoglobulins. These contribute to the globulin fraction of plasma proteins. They are soluble proteins secreted by the B cells and the plasma cells. The antibodies, they contain 4 polypeptide chains in which there are 2 identical low molecular weight chains and 2 high molecular weight chains. They are linked by a disulfide bond. Now both chains have a constant and variable regions. There is one region which is constant, that is uniform, and one region is variable. This variable region has unique amino acid sequence for a specific individual immunoglobulin, because that will attach to a specific antigen. Therefore, the variable region of the chain is important in identification of the particular antigen. Therefore, for a particular antigen, particular antibody is produced. Now, types of antibodies. There are 5 types of antibodies according to their number in the blood or serum. They are given here. starting from large number to lesser number. Number 1 is the IgG. It is present in maximum number, that is about 1000 mg/cent. They are important in primary and secondary responses. They can cross placental barrier and provide passive immunity to the fetus. Second one is the IgA type of antibody. It prevents attachment of pathogens to the epithelial cells. Epithelial cells of the body, they prevent attachment of these antigens. So their number is about 200 mg/cent. The third type of antibodies are IgM. They are released as primary responses from plasma cells. Their number is 120 mg/cent. The fourth one is IgD. It attaches to B cell and activates it. Their number is only 3 mg/cent. And the least number of antibodies found are those of IgE type. These binds to mast cells and basophils to cause histamine release. You know, when there is a allergic type of reaction, these antibodies helps in production of histamine to evoke a allergic reaction. Now, functions of antibody. As such, antibody do not destroy the antigen on its own, but it helps in destruction of antigen indirectly. It forms an antibody-antigen complex and acts through first neutralizing the antigen, that is pathogen, then agglutination of the antigen. By agglutination also the pathogen is destroyed. Then after agglutination, precipitation of this complex. Then with the help of complement fixation, that is by secretion of enzymatic proteins that kills the antigen. Now here in this picture, you can see the antigen-antibody complex reaction. It is summarized here. The antigenic reaction with antibody, this forms a complex which activates agglutination, neutralization and precipitation. Now this promotes, when the antigen is neutralized, agglutinated and precipitated, then comes the role of phagocytes. The phagocytes finally destroys them. Then in such cases, if the infection is more, then a process of inflammatory action may be provoked. Here also, lot of neutrophils comes out of the blood vessels and they phagocytize the infected cells, the antigens finally. Now here in this case, this formation of complex activates the complement factors. These complement factors produces proteins. That protein ultimately kills the pathogenic organisms. So that is how the whole antigen-antibody complex reaction occurs in our body. Now coming on to the cell-mediated immune response once again. The cell-mediated immunity is needed for intracellular antigens. because in this case antibodies are not effective, because antibodies will not enter into the cells. T cell mediates cellular immunity through, there are two ways in which the T cells mediate the immunity. One is T4 cells, which contain CD4 protein. These are called helper T cells or TH cells. Secondly, through TH cells, which contain CD8 protein, there are cytotoxic T cells, which destroys cells containing foreign antigens. Other T cells are suppressor T cells and memory T cells. Suppressor T cells are those cells which suppresses the immune action, whereas the memory T cells retains the memory for immune action. Now, activation of T cells. The T cells are activated, they forms antigen receptors, that is called T cell receptors, which binds to MHC protein complex. We have already studied it earlier. Then these T cells, they form clones. So many T cells are formed by cloning. It is stimulated by certain cytokines and interleukins. Now, MHC, that is major histocompatibility complex, are of two types. We have already discussed is MCH1 is found on all the cells except RBCs. The endogenous antigens, that is viral and cancer, stimulates CD8 cells, that is T8 cells population and cytotoxic T cells. Then MHC2 found only in the plasma membrane of B cells, plasma cells and APCs, that is antigen presenting cells, in response to the exogenous antigens. CD4 population of cells stimulate to produce helper T cells, those are called as T4 T cells. Now, here in this picture, you can see the cytotoxic T cell actions. So, first of all, whenever there is an exposure from antigen, antigen is received here by the T cell, then a MHC1 is formed, it is attached to T cell receptor. Some of these cells form memory cells, which retains memory for the future. The other cells, they become cytotoxic T cells, that is T8 cells. These attacks the target cells and kills them ultimately. This is how the cellular immunity actions through cytotoxicity. Now, functions of different types of T lymphocytes, shortly called as T cells. One is T helper cell. It regulates immune function by secreting lymphokines. Lymphokines include interferon G, monocyte colony stimulating factor, and interleukins numbered as 2, 3, 4, 5 and 6. It stimulates growth and proliferation of suppressor and cytotoxic T cells, both suppressor cells and cytotoxic T cells via interleukin 2. They also stimulates B cell growth via interleukin 4, 5 and 6, and proliferation of plasma cells and antibody formation. They stimulate phagocytic activity of macrophages for destruction of antigens. Now, second one is cytotoxic T cells. As we have already discussed, these are the cells which destroys the pathogen. These directly kills the organisms or cells binding with antigen. After binding with target cells, they release perforins. As we have already discussed, these form pores in the cell membrane of the pathogen. Then granzymes they secrete. These granzymes destroy the cellular component, thereby destroying them finally. Cytotoxic T cell destroy foreign antigens, cancer cells. They destroy foreign antigens as well as cancer cells. Then there are certain suppressor T cell. You know, when these suppresses both helper T cells and cytotoxic T cells. These are called regulatory T cells because they prevent the damage of own tissue. You know, when an immune action is generated into body, say suppose for example, if a virus enters in our body, the system identifies the virus infected cell. But in case if the proliferation of these cells, especially helper T cells and cytotoxic T cell, increases abnormally, then it will destroy the cell, normal cell of the body. To prevent that, suppressor T cells are there, which prevents the growth of production of the cytotoxic T cells and helper T cells. So, therefore, they are called as regulatory T cells. Now, coming to the final part and important part of this topic, that is immunological disorders. The first one is SCID. Mostly immunological disorders are those disorders, those diseases which are due to our improper or defective immune response. Number one is SCID. This is called as severe combined immunodeficiency syndrome. When we talk about syndrome, in syndrome there are many symptoms of a disease and it is called as symptom. It is a genetic disease in which results in deficient B and T cells. Number of B cells and T cells is reduced. It is fatal if untreated. For treatment, bone marrow transplantation is done. The example of SCID, one of the example of SCID is Hodgkin's disease. Now, the second and most popular example of immunodeficiency is the AIDS, that is acquired immune deficiency syndrome. It is caused by a virus called human immunodeficiency virus, the HIV virus. It affects by interfering the activity of helper T cells, that is T4 cells. Now, how the AIDS is transmitted? The transmission of AIDS is the main problem of this disease because it is mainly transmitted by so many methods. It is transmitted by infected blood transfusion. It is transmitted more commonly by unprotected sex, sex between heterosexuals, sex between homosexuals, anal sex, oral sex and so on, by using infected needles, especially in case of drug addicts uses common needles amongst themselves. So that is also one of the common reasons of infection. Then this infection crosses the placental barrier, the virus crosses placental barrier. So the fetus gets the infection from the mother. It can also be spreaded by the breastfeeding. Then symptoms. These symptoms of AIDS, they are common disease symptoms. So it is very difficult to identify it at early stage because the period of appearance of severe symptoms is very long, sometimes in years. So the symptoms include fever, headache, fatigue, lymph node enlargement, that is main, vomiting, diarrhea. These are all common symptoms. Patients are at the risk of developing cervical cancer, Burkitt lymphoma, etc. Treatment is very general. There is no specific treatment. Antiretroviral therapy is given for the treatment. Now coming on to allergy. Allergy is a hyperactivity response to an antigen which is usually tolerated by others. You know, it's not necessary that one agent which causes allergy to a person will be allergic to others also. So, if it is allergic to one person, it may not be allergic to others. Two types, it is local and systematic. If it is localized at one part of the body, it is called as local allergy. It is confined to an area of the body like eczema, urticaria, hives, swelling on lips, etc. So this is a kind of allergy which is called as local allergy. But if the allergic reaction is widespread throughout the body, then it is called as systemic allergy, or in technical term, it is called as anaphylaxis. This is a severe kind of allergic reaction. It is a generalized allergy affecting systemic functions. It is an acute medical emergency. May lead to shock. Sometimes it may lead to death. Anaphylactic shock, it is called. Example, after injection of penicillin and xylocaine. You know, both these drugs are very common in the therapy. Penicillin is an antibiotic which is very commonly used, but for some people it is allergic, and sometimes injection of penicillin becomes fatal because it provokes anaphylaxis. Similarly, xylocaine is given for anesthesia of local site, especially in case of tooth extraction, in case of small surgeries, xylocaine is given. But the reaction of xylocaine is very severe. Therefore, for such type of allergic drugs, there should be a sensitivity test before administration of such type of drugs. Now, hypersensitivity reactions are of two types. One is, just I explained, type 1 is anaphylaxis. It's a severe kind of action. It is IgE mediated. IgE antibodies bind with mast cells and basophil cells, and these two types of cells releases large quantity of histamine. Here it is, which is a vasodilator. Now, local effects include runny nose, watery eyes, itching, and redness. Histamine produces bradycardia and hypotension. Bradycardia means lower heart rate, and hypotension means low blood pressure. Then type 2 cytotoxic reactions, these are caused by IgG type of antibodies and IgM type of antibodies. It binds with the antigens present in the body cells, especially RBCs. So, this kind of allergic reaction is seen in cases of mismatched blood transfusion. If mismatched blood transfusion is there, there is an immediate allergic reaction which can be fatal to the patient. So, that is caused by the IgG and IgM antibodies. Type 3 is the immune complex diseases. Antigen acts with antibody forming antigen-antibody complexes. These are not destroyed and remain attached to the basement membrane of blood vessels, causing inflammation. Such type of allergic reaction is seen in glomerulonephritis of kidney, then rheumatoid arthritis. In the fourth type, hypersensitivity reactions, it invoke helper T cells and cytotoxic T cells and macrophages to great level, maximum level, which becomes harmful to the patient, as in case of the test of tuberculosis, Tivitin test, in case of heavy metals. Then bronchial asthma, another very common example of allergy. The allergens causes release of anaphylactic substances from the bronchioles, causing bronchospasm and breathlessness. Especially in the season when there are a lot of pollen grains in the air, many of the peoples are allergic to those pollen grains, causing asthmatic hyperactivity reactions. Now, one last part of this topic is the organ transplant. As you know, when some vital organ is affected by a disease, then for survival of that person, organ transplant is done. It is very common as a skin transplant in cases of, especially in cases of burns and major injuries. Skin has to be transplanted. Kidney failure is very common these days because of the consumption of various type of drugs which are metabolized in the kidney, which affects the kidney functioning. Then liver damage is also has become very common these days. In case of bone, if there is a damage of bone, the bone has to be replaced. So, the common transplanted organs include skin, kidney, liver and bone. The recipient develops immune response against the transplanted tissue which is foreign to them, because each and every individual has got some kind of proteins, antigens, which are specific to him only. So, when the tissue or an organ from a person is transplanted to another person, it evokes a rejection reaction, an immune reaction by which the transplant is rejected. Now, transplant rejection occurs due to activation of cellular immunity. Because the transplanted tissue is foreign, so the cellular immunity is developed against it and it is rejected. Now, types of grafts or types of transplant. Number one is autograft. the safest one, the transplant from one part of the body is transplanted to the other part of the same person. It is the safest because the genetic composition is the same, the antigens are the same, so there is least possibility of rejection. It is commonly seen in case of skin transplant. If the skin of one place of the body is damaged, then the skin from other place of the body of same patient is transplanted like that. Then second one is essograft, transplant between identical twins. As you know, the identical twins develops from the single embryo, so they have the same composition, genetic composition, they have the same antigens in their body, so there are least chances of transplant rejection. Third one is allograft. Allograft means the transplant between the individuals of same species. Within the same species, majority of the genes, majority of the antigens are similar, so the transplant is successful to some extent. This is a transplant being taken from the close relatives, the first cousins or brothers and sisters, from mother to child, from child to mother, from father to son, and so on. Then the next category is xenograft, where very rarely it is done. It is taken from another species. Sometimes when there is a mass destruction of skin due to burns, then animal skin is used temporarily for wound healing. In such cases, like burns, it is used. Its rejection is very fast, because by and large, the antigens of recipient and donor species are entirely different. Now, for treatment of the transplanted tissue or grafts, the immunosuppressive drugs are given so that the immune response is checked to prevent the rejection for a longer period, like azathioprine, glucocorticoid therapy is used, which suppresses cytotoxic T cells. Then monoclonal antibodies produced against T lymphocytes and antibodies, they are used. Cyclosporins, rapamycin, these are used as for the treatment of transplant rejection. Now, monoclonal antibodies, this is a term, this is a technology which is used at a very large scale these days. Monoclonal means when the antibody producing cell is cloned to produce same type of antibody having the same composition, those are called as monoclonal antibodies. The procedure is like that: antibodies prepared from a single B cell or plasma cell produced by fusion of B cells with a tumor cell that finally produces clonal antibodies in large number. You know, we have seen that when a particular antigen enters in the body, the B cell produces particular antibody for a particular antigen. Now, if that B cell is somehow cloned to produce plasma cell, or plasma cell is cloned, then you can produce similar kind of antibodies which are called monoclonal antibodies. Now, how it is done? We take the B cell, which is activated to produce specific kind of antibodies and fuse it with a myeloma cell, that is a cancer cell. You know, the cancer cell has got a tendency to multiply abnormally in large numbers. So what we do is that we fuse the B cell activated for a particular type of antigen with a myeloma cell, that is cancerous cell, and this is called hybridoma. This hybrid cell, which has got a characteristic of cancer cell, is called hybridoma. This hybridoma produces similar kind of antibodies in huge numbers. Those are called as monoclonal antibodies. These monoclonal antibodies are used in a very large scale for production of vaccines. They are also used for treatment of autoimmune diseases, for detecting various kind of diseases, especially cancer in early stages. They are used for identification of allergic diseases. They are also used for identifying different kind of sexually transmitted diseases. Now, autoimmune diseases. There are certain situations when the immunity of a person becomes harmful to himself. Immune system triggers immune response against the body of the same person. So, common examples are SLE, that is systemic lupus erythematosus, then rheumatoid arthritis, myasthenia gravis, very popular it is, then Graves' disease, psoriasis, again very common autoimmune disease of skin, then type 1 diabetes, it is diabetes since birth. So, all these are autoimmune diseases. That's all for today. Thanks.